Hello, could somebody confirm you can hear me?
Yeah.
Thank you. All right, are there any questions? That's not the right one, is it? Yeah, it is. Okay.
I had a question about the quiz times, whether it was going to be applied for this quiz.
I think I've got the discussion open. Let me close this down and look into that. I think the discussion is open until tonight or tomorrow, in which case it will not affect the quiz. I haven't looked at that discussion. Could anyone tell me that they've looked at it and what the comments were?
I think that everybody put 11:59.
Okay. Like I said, I haven't looked at that. I will look at that and we'll let you know. But like I said, I think the discussion is still open. I'm trying to look now. Um, week 4. There it is. Week 4 lecture. Come on. It's not showing me what the end date is on this. May 3rd. So it's still open until May 3rd. If you haven't given a comment about what you would like to see for the quiz, please put it in the quiz discussion. And it'll close tomorrow. All right, let's go back. Let me share my screen. All right, you should be able to see my screen now. Today is May 2nd. And we are supposed to be discussing chapter 5. Did we finish chapter 4? Does anyone remember? Let me take a look at that. It looks like we may have been to here. Let me check. Oh, that's not right. All right, this looks like this is the last slide we discussed. Is that right?
Yes.
So we discussed endospores. The next section is on eukaryotes. Eukaryotes would be a repeat of material you've had in Biology 160. So I'm not going to cover this material. I do need to talk about the endosymbiotic theory, though. Oops, went too far. There we go. So the endosymbiotic theory is a theory to explain the evolution of mitochondria and chloroplasts in eukaryotes. There's a question about how these organelles originated. And these two organelles are different than most other organelles in the cell. For one thing, they have a double membrane. They have DNA inside them. And there's a few other differences as well, and we'll talk about some of these differences. And you can read about this on chapter 10, page 275, page 276 in the third edition of the custom textbook. All right. The endosymbiotic theory, let me blow this up, proposes that there was a precursor pre-eukaryote. So I'm calling it a pre-eukaryote, an ancestral eukaryote, and it engulfed a prokaryote. So let me go ahead and draw that. Actually, let's see if I can get paint open. Let's blow this up. So we had a very large cell, which is our precursor to a eukaryote, meaning it's before eukaryotes were around. And it lacked mitochondria and chloroplasts. Obviously, mitochondria are found in animal cells and both are found in plant cells. This cell engulfed a bacteria. And for whatever reasons, when it engulfed it, brought that inside the cell, it didn't immediately eat it. So let me get the eraser now and get this ready. And when it engulfed it, it would make a vacuole around it. So it would have two membranes because it was brought in by pseudopods, and then this became a vesicle being brought in the cell. And then there would be the cell membrane of the bacteria. So it'd have two membranes. And for whatever reason, it didn't eat the bacteria immediately. Now, this precursor eukaryote could make ATP from fermentation. So it could take one glucose and make 2 ATP per glucose. Any questions about this? Oops, let me go to capital. The bacteria could engage in aerobic respiration. So this is the bacteria. And blow that up. Let me make it longer. And it could take one glucose and get about 38 ATP per glucose. For whatever the reason, this bacteria took some glucose from the cytoplasm of the proto-eukaryote, and for whatever reason, made the 38 ATP and shared some of that ATP with the proto-eukaryote. Why that might have been the case is simply that life was primitive and early back then, and it just floated out. And what am I saying? The proto-eukaryote shared some of that ATP with, the bacteria shared some of that ATP with the proto-eukaryote. And the proto-eukaryote then, I'll make another one here. wondered what was going on. Oops, I lost one ATP. Not one ATP, one glucose, sorry, one glucose. But gained many ATP. And we don't know how much ATP was initially shared. It probably wasn't all 38 of the ATP that were made, because some of the ATP would have been used by the bacteria. But the point is, it was much more than 2 ATP. And so the proto-eukaryote decided to keep this bacteria around because the bacteria was making ATP in greater numbers than the proto-eukaryote could. Any question about any of that? So the two lived in symbiosis. That's lived together. The proto-eukaryote gave the bacteria a home and glucose, and then the bacteria made ATP for the proto-eukaryote. Any question about that? Now, the two have lived together for so long now, you find almost all eukaryotes with the bacteria in them. And the bacteria is actually no longer free living, that we only find it inside of the cell of the proto-eukaryote. Any question about any of that? And the bacteria evolved into the mitochondria. Let's bring it over here. And the proto-eukaryote is now the eukaryote. And we can say that because it has, let's blow this up a little. Because it has a mitochondria. Okay, so this is the theory of how the mitochondria evolved. And it's also the theory how chloroplasts evolved, and I'll talk about that in just a little bit. Any questions about this theory? All right, let's discuss some of the evidence for this theory. And in animal cells, this happened one time. Let me state, in plant cells, this happened 2 times. The mitochondria came into the bacteria came into the cell, which then evolved into becoming the mitochondria. And then another photosynthetic bacteria came into the cell, and it evolved into the chloroplast. There's strong evidence for this theory. First, when we look at the few eukaryotes that lack a mitochondria, all of them are primitive amoebas. What they do is they will engulf bacteria, which can engage in aerobic respiration. And then the eukaryote, which is an amoeba, can only engage in fermentation to make ATP, but the bacteria can engage in aerobic respiration. And so the amoeba feeds the bacteria glucose and gives a home to the bacteria. And then the bacteria uses up that glucose to make ATP and then releases some of that ATP to the amoeba. Here we're actually seeing another piece of evidence of the endosymbiotic theory, where this protist, this looks like a paramecium, has engulfed a bacteria which is green because it engages in photosynthesis. And it looks like here the bacteria is about to split into two cells. But the point is that the paramecium is working in symbiosis with another organism, a prokaryote, and the prokaryote is making, in this case, food for the paramecium. Another piece of evidence is that the mitochondria and the chloroplast have two membranes. They have an outer membrane, which would be the membrane of the vesicle, and they have an inner membrane, which would be the cell membrane of the bacteria. Any question about that? No, let me see where my... There it is. Some evidence, other evidence for it is when we look at the DNA of prokaryote cells, the DNA in a prokaryote cell usually has one circular DNA molecule, usually one. The eukaryote has usually many linear DNA molecules. The mitochondria and chloroplast DNA is one circular molecule. In prokaryote cells, the DNA lacks histone proteins in, I should say not prokaryotes, but in bacteria. And the eukaryotes have histone proteins on their DNA. The mitochondria and the chloroplast do not have histone proteins on their DNA. When we look at the first amino acid synthesis, first amino acid made in protein synthesis, as you know, in eukaryotes, the first amino acid that AUG codes for is methionine. AUG in a prokaryote cell does not code for methionine. It codes for formal methionine, a similar but different amino acid. When we look at the first amino acid coded for by AUG in mitochondria and chloroplast, it's formal methionine. When we look at the ribosomes that are made in a eukaryote cell, meaning the DNA encodes these ribosomes, they're 80S in size. When we look at the DNA encoded by a prokaryote cell, the genes code for 70S ribosomes in size. And when we look at the DNA of mitochondria and chloroplasts, they do have the genes for a ribosome, but that gene codes for a 70S ribosome. Furthermore, when we look at how the cells grow, prokaryotes mostly divide by binary fission, meaning that's how the cells grow. Eukaryotes grow by mitosis, and mitochondria and chloroplasts grow by binary fission. There's a few other pieces of evidence, like we could say that the DNA of a eukaryote cell, the genes have exons and introns, and in bacteria, they do not have introns and exons. On the mitochondria and chloroplasts, they do not have introns and exons. But the strongest piece of evidence for the mitochondria and chloroplast belonging to the bacteria is that when we sequence the DNA of any organism that's a eukaryote, the DNA is similar to other eukaryote DNA. And then the same is true when we sequence the DNA of a bacteria. The sequence of DNA will be similar to other bacteria. When we sequence the DNA of a mitochondria in the chloroplast, the DNA is similar to the DNA of a bacteria. So that's the evidence in favor of the endosymbiotic theory, and that is in animals, an early precursor animal cell took in a bacteria which evolved into the mitochondria, and we don't find that bacteria free living anymore. It's only found in eukaryotic cells. And then plant cells did this twice. They took in a bacteria that evolved into the mitochondria, and then they took in a second bacteria which evolved into the chloroplast. Any questions about any of that? All right, that's the endosymbiotic theory. If there's no questions, let's move on to our next lesson, chapter 5. Let me remind you that the quiz this Wednesday will be covering chapter 4, the end of chapter 3, and then labs 5 and 6. We will be discussing the unknown project in the lab today. All right. Any questions about what we're going to do? So chapter 5 covers microbial metabolism, as is normal. I have the major goals and the rough outline listed here. Know the terms. We'll talk about each of them. Understand what feedback inhibition is, and understand what aerobic respiration is, its steps, and how aerobic respiration differs from anaerobic respiration. Any question about what we'll be covering? All right. Microbial metabolism is all of the chemical reactions occurring in a cell or an organism. And when we're talking about microbiology, usually most organisms are one cell. So all of the chemical reactions that are occurring in a cell. We can break metabolism into two parts, catabolism and anabolism. Catabolism is the decomposition reactions where you start with a larger molecule and you break it down into a smaller molecule, such as breaking down starch into glucose. Catabolism does release energy because the larger molecule stores more energy than the smaller molecules. Any question about any of that? All right. Anabolism is the biosynthesis reactions, and they start with smaller molecules. You bring them together, and then you make a larger molecule, such as taking glucose, hooking them all together, and then making starch. Anabolism requires the input of energy because the larger molecule has more energy than the smaller molecules. Now, catabolism and anabolism are similar to, oh gosh, what's the name of that? I had it on the tip of my tongue, now I've forgotten. Exogonic and introgonic, is that the term? Let me do a search there. Oh, there we go. Oh, endergonic, exergonic, and endergonic. In an exergonic reaction, energy is given off by the reaction. In an endergonic reaction, energy needs to be supplied into the reaction, or else the reaction cannot proceed. The point is that catabolism is looking at the size of the molecule, and an exogonic reaction is looking at the energy given off by the reaction. And then an endergonic reaction is looking at the energy being taken into the reaction. And anabolism is looking at the size of the molecule, where smaller molecules are made to make larger molecules. Usually catabolic reactions are exergonic and anabolic reactions are endergonic. Any question about any of that? All right, this reaction is showing you how metabolism can be hooked together, and that is you start with a complex molecule like starch, and in a catabolic reaction, you can make the simple molecules like glucose. In the catabolism of starch by aerobic respiration or fermentation, you can generate ATP. But please understand that human cells, and actually all animal cells, are lousy at making ATP, and that we only have two reactions that are catabolic that can make ATP, and those are fermentation and aerobic respiration. Okay, and so those are the only two reactions that can make ATP. All other catabolic reactions do not make ATP. Like when we take sucrose and break it down into glucose and fructose, that is a catabolic reaction, but it does not generate ATP. Any questions about any of that? All right, whenever energy is moved from one form or another, such as a complex molecule like starch, and put it and is changed to the energy in another molecule like glucose, whenever energy is changed from one form to another, some of that energy is lost as heat, and heat is another form of energy. So you can actually just say some of the energy is converted into heat. So in catabolic reactions, heat is always given off. Some of that energy in some of the catabolic reactions, and in animal cells, it's only two of those catabolic reactions. We can convert some of that energy into ATP. And how you do that is you join ADP and phosphate, and you add in the energy from either aerobic respiration or fermentation, and then some of that energy will be stored in ATP. Some of that energy is lost as heat, and then of course some of that energy is converted from the complex molecule like starch into the simple molecule like glucose. Any question about catabolism? If not, let's go on to anabolism and state that simple molecules are made into a larger molecule like glucose being made into starch. That reaction cannot occur unless energy is put into the system, because starch contains a lot more energy than glucose. How the cell normally gets its energy to convert glucose into starch or a simple molecule into a larger molecule is it normally gets the energy from breaking down ATP, and that releases a lot of energy. ATP breaks down into ADP and phosphate, and some energy is given off. Some of that energy can be used to make the complex molecules. Now, whenever you convert one form of energy to another, such as the energy in glucose to the energy in starch, some of that energy is given off by heat. So in an anabolic reaction, you also have heat that is created or given off, even though energy is needed for this reaction to occur. Excuse me. That energy is always given off when you're converting energy from one form to another is known as the second law of thermodynamics. Any question about anything on this slide? Remember, not all catabolic reactions make ATP, and in animal cells, only 2 do. Green plants do make ATP in other ways. They're much more efficient than animal cells, but even green plants, the majority of their catabolic reactions, and we're talking about the vast majority, do not make ATP. And humans and animals only have 2. All right. So when we're talking about chemical reactions, you can hook chemical reactions together to form a metabolic pathway. So this is one chemical reaction where molecule A is converted by enzyme 1 into molecule B, molecule A being the starting substrate of the metabolic pathway. And then molecule B can be converted into molecule C by enzyme 2. So that's the second step in the metabolic pathway. And then molecule C can be converted into molecule D by enzyme 3. And then molecule D can be converted to molecule E by enzyme 4. And then lastly, molecule E can be converted into molecule F by enzyme 5. Molecule F is the end product. It's the last product made in this metabolic pathway, so we call it the end product. All of these other molecules are called intermediates or intermediate products because B is the product of this one reaction, converting molecule A, the starting substrate, into molecule B. Metabolic pathways are determined by enzymes, and enzymes are encoded by genes. So metabolic pathways are determined by your genes or an organism's genes. Any question about any of that? All right, this slide is showing you all of the metabolism we need to discuss and you need to have memorized. Are you ready to begin? That's a little joke. Obviously, we're not going to discuss this much metabolism, but this is all of the metabolism that we know of that is going on in a single cell. All right, let's talk a little bit more about chemical reactions. Chemical reactions occur according to the collision theory, and they occur as atoms, ions, and molecules collide with each other. And if they have enough energy in the collision, they will then be converted into the product of this chemical reaction. The activation energy is the energy needed to disrupt the electronic configurations in the original atoms or ions or molecules that are colliding and then convert these atoms, ions, or molecules into the product. So the reaction rate is the frequency of collisions with enough energy to bring about a reaction. So if two atoms have enough energy and they collide, they will, by the collision theory, be changed into a chemical reaction into the product. You can increase the reaction rate by increasing the abundance of enzymes. More enzymes, you can speed up the chemical reaction, meaning the enzyme will bind to the atoms and make them into products. And you can also increase the reaction rate by increasing the temperature. Temperature is the movement of molecules or atoms or ions. And the higher the temperature, the more the starting substrates move about. And the more they move about, the more energy they have. And the more energy they have, the more likely they're going to have the activation energy to proceed with the chemical reaction. So increasing the temperature will increase the rate of a reaction, and then increasing the pressure does the same thing. Increasing the pressure, you can think of it as getting a box and shrinking it or something. It puts the molecules more closer together, more likely to collide, and then more likely to collide with enough energy so that the chemical reaction can proceed. I won't ask you about the pressure because it's kind of difficult to conceive, but think about, I don't know, a chemical reaction happening on the surface of the planet Earth. It's 1 atmospheric pressure. And then you think about it happening on the surface of Jupiter, which doesn't have a surface. So we'll just say the start of the liquid area, which we're not even going to get into that. But the point is that it's under much more pressure. And so the reaction is much more likely to proceed, assuming the molecules are starting with the same energy, the same movement, the same temperature. Okay, because of increased pressure, the reaction rate is going to proceed at a greater pace. Any question about any of that? All right, enzymes reduce the activation energy. So here we have the energy on the y-axis and just the time on the x-axis. And this we have actually, I guess that's one molecule, which is going to break apart into products, 2 products, which I can't read that. Products A and B. The initial molecule is larger, it has more energy than the end products. So energy will be given off by this reaction. The reaction is likely to proceed because the energy is going downhill, meaning it's a catabolic reaction. However, to convert the starting reactant to the products, you do need to add energy before molecule AB, meaning the initial reactant, can be converted into the products. An example of this would be something like sucrose. We could split sucrose into glucose and fructose, and that reaction would proceed because it's downhill. And energy is given off, and that's the energy given off will be the difference between the initial energy level and the ending energy level, which isn't shown here. However, to convert sucrose into glucose, it doesn't happen very often. And you know that because you put a cup of sucrose, which is regular table sugar, and you leave it on the counter. It doesn't convert into glucose and fructose overnight. I mean, it would take centuries actually to happen, but it would slowly happen. And what would happen is actually probably, what do you call that? A cosmic ray from outer space would come and collide with molecule AB, the sucrose, and that would give it enough energy to get over the energy hump and then become glucose and fructose. Like I said, it's not going to happen even overnight. It's not going to happen. It's not going to happen for centuries. Okay, meaning not one, but the whole cup. It would take centuries, maybe even longer, like millennia, to convert from sucrose into a cup of glucose and fructose. The reason is because the initial reactant needs to get energy to get over the reaction rate or the reaction without an enzyme. And that is called the activation energy without the enzyme. And that's actually quite high. If you took a blowtorch to the sucrose, well, that's actually not burning it into glucose and fructose. That's actually burning it into CO2 and water, but that would be similar. But that's just showing you, let's convert this instead of saying sucrose into glucose and fructose. Let's say glucose, and we're breaking it down into CO2 and water. How you do that is you take a blowtorch to it, and you know that's like 1,000 degrees Fahrenheit, and that gives it the energy it needs to get over the energy hump of the activation energy without an enzyme. And then you would burn all of that glucose into CO2 and water. Any questions about that? I switched it because it's easier to talk about and easier to understand. However, glucose is in your cells being converted into CO2 and water all the time. And our cells actually burn glucose and burn it down into CO2 and water. And we're not taking 1,000 degrees Fahrenheit because that would fry your cells. It's actually only 37 degrees Celsius. That is called the activation energy with an enzyme. And this scale isn't to scale, meaning this graph isn't to scale, I guess. And that is, this is 37 degrees and that's like 1,000 degrees. So the point is the activation energy without an enzyme is much less than the activation energy without the enzyme. And in our cells, we convert glucose into CO2 and water all the time. All right, any questions about any of that? All right, enzymes are biological catalysts. They can speed up a chemical reaction 10 to the 8th to 10 to the 10 fold times. A catalyst is just a chemical that can speed up a chemical reaction. The chemical reaction is where you change one molecule into into another molecule, like converting glucose into CO2 and water. Biological catalysts or enzymes are encoded by genes, and so our genes determine which chemical reactions we can engage in. Enzymes are specific for a single chemical reaction. That's not entirely true. As you know, we have different drugs which can mimic the molecule the enzyme is working on. But for our purposes in this class, we can say it's close enough that an enzyme is specific for one single chemical reaction, converting one molecule into its products. And another molecule would require another enzyme. Any questions about any of that? For example, breaking sucrose into glucose and fructose. We have a specific enzyme that we call sucrase. And then to convert lactose into its monomers, we have another enzyme called lactase to break down that molecule. And those molecules are very similar. The specificity of enzymes is due to the enzyme's three-dimensional shape. Each enzyme acts on a specific biochemical reaction, and each enzyme acts on a specific reactant, also called the substrate. Enzymes are further not used up or altered in a chemical reaction, so after they performed one chemical reaction, they can bind to the other substrate, another substrate, and then perform that chemical reaction countless times. Any question about any of that? Furthermore, enzymes form an enzyme substrate complex, which they do when they're performing the chemical reaction. This enzyme substrate complex orients the substrates to increase the probability of a reaction, and this lowers the activation energy. What's happening is the enzyme substrate complex starts putting stresses on the substrate, the bonds of that substrate, and that will help convert the substrate into the product. You should know that enzyme names usually end in "ase." We'll talk about some of these. You don't need to know any of these, but we will talk about ligase, and I think we talk about transferase. I'm not sure if we talked once about oxyreductase, but you don't need to know any of these examples. But you should know that most enzyme names end in "ase." That isn't true for all enzymes, especially the older enzymes that were named before this convention came into effect. Like renin is a enzyme in our stomach. It's important in babies. It curdles the milk and then keeps the milk in the stomach longer so it can be digested better in the stomach. You should also know that enzymes do not act in a vacuum. Some enzymes require a helper, and these are called coenzymes if the helper is a biological molecule, or the helper is called the cofactor if the helper is a non-protein portion, a molecule that activates or helps activate the enzyme. And usually the cofactors are simply minerals or elements like iron, zinc, magnesium. A coenzyme is a biological molecule like NAD plus. We'll talk about NAD plus in aerobic respiration. And the coenzyme actually binds to the enzyme and then makes a spot for the substrate to then bind to the, enzyme and coenzyme. You don't need to know the names, apoenzyme, the enzyme before combining with the coenzyme, and the halo enzyme, the whole enzyme. You don't need to know those names. There are some important coenzymes that we're going to discuss. you do need to know the first two because we're going to discuss these two in aerobic respiration. But NAD plus is an important coenzyme, and its long name is nicotinamide adenine dinucleotide, which probably means as much to you as NAD plus. Another important coenzyme is FAD, flavine adenine dinucleotide. You need to know these two because we will discuss them in aerobic respiration. There are other important coenzymes like NADP plus, which is found in, like green plants, FMN, and then coenzyme A, which we actually do talk about coenzyme A, but not as a coenzyme. Anyways, only know the first two. We will discuss them in aerobic respiration. This slide is showing you how an enzyme actually binds to the substrate. And then they find the enzyme substrate complex, which puts stress on the chemical bonds of the substrate, making it easier to convert the substrate into the products. And once the product is made, it then comes off the enzyme. The enzyme is now free, and it can come and bind to the substrate again and again and again. This top model is showing you the lock and key model, where one substrate fits one enzyme, similar to one key fits one lock. Now, the lock and key model of an enzyme is a good model, but it's a little oversimplified because the lock and the key model does state that the substrate binds to the active site of an enzyme, forming the complex, which then converts the substrate into the product. The product comes off, freeing the enzyme to bind again. But it's a little oversimplified, and the next model of an enzyme is showing you the hand in the glove model, where the enzyme binds to the substrate in its active site, right there, and then that puts stress on the substrate, converting it into the product. What do you notice about this picture? There are three things you should notice. Let me blow that up for some people there. Anyone notice anything? One's very easy. Look at the size. Nobody's going to comment. I'm not going to comment if nobody comments.
Does the enzyme kind of close around the substrate?
Yeah, actually, that's why they call it the hand in the glove model. And that is the enzyme changes its shape to fit the substrate. So the enzyme is changing its shape, sort of like the glove, changes its shape to fit the hand. And that's the first of the three parts of the hand in the glove model. The other, which is quite obvious, and I'm surprised nobody's mentioned it, is the enzyme is much larger than the substrate. These enzymes are very large molecules compared to the substrate that they bind to. And they bind at the active site, which is shown here in the hand in the glove model, or there in the lock and the key model. The third one is hard to see. You have to look really closely at the substrate here and the substrate there. And you'll notice that this substrate is a little flatter than this substrate. So even the hand in the glove model is incorrect. It should be said something like the shapeshifter's hand going into the glove, where the glove molds to fit the shapeshifter's hand, but the shapeshifter's hand is also molding to fit the glove. And those of you who are familiar with Star Trek Deep Space Nine, you know what I'm talking about when I'm saying shapeshifter's hand is molding to fit the shape of the glove. Any question about that? All right. Enzymes do not work in a vacuum. I've already stated that. They do require the correct temperature, the correct pH, the correct salinity. Generally, they require the absence of heavy metals, alcohols, UV radiation. All of these substances can damage the protein of the enzyme, causing the protein to denature. If you have the enzyme at the incorrect temperature, the enzyme will lose its shape or start losing its shape, take on a different configuration, and then we call it denatured. And that's true at the wrong pH, the wrong salinity, in the presence of heavy metals, in the presence of alcohol, and in the presence of UV radiation. The protein can change its shape, begin to unravel, and we call that beginning to denature. And then if the temperature is way off, or the pH is way off, or the salinity is way off, heavy metals are in high abundance, alcohols in high abundance, the protein will totally denature and have no function. So the enzyme won't work. And with temperature for our enzymes, which work best at 37 degrees, that would happen around freezing temperature or a very warm temperature, which I'm not sure how warm, but somewhat less than boiling water. So somewhat less than 100 degrees Fahrenheit. Sorry, 100 degrees Celsius. That would be 212 degrees Fahrenheit. Okay. The substrate concentration can also influence the enzyme's activity. At least at the start, when the substrate concentration is very low. When you increase the substrate concentration, you will increase the amount of enzyme activity. Various enzyme poisons can also affect the activity of the enzyme. Poisons usually, well, not usually, poisons always decrease the amount of enzyme activity. If the poison is affecting the enzyme, you can totally denature the enzyme, in which case you'll have no enzyme activity. Two poisons are cyanide and fluoride. You don't really need to know those, but just giving you some examples. We'll talk a little bit more about this in future slides. Enzymes don't work in a vacuum. They can be inhibited. We can have two types of inhibition: competitive inhibition and non-competitive inhibition, or allosteric inhibition. And then lastly, we'll talk about feedback inhibition and how it can be used to turn off and turn on a metabolic pathway. Any question about any of that? All right, so we have our normal active enzyme under normal conditions, like normal temperature, the optimal pH, the optimal salinity. You put it in a suboptimal environment, one that is either too hot or too cold, the protein will take on a different shape. It will no longer be supercoiled, and let's say the active site's right here. That active site now is, I don't know, someplace all over here. and it cannot bind to the substrate. So that protein would be totally denatured and would not have any function. And for like temperature, that would be definitely at freezing temperature for our enzymes, and definitely at boiling temperature for our enzymes. And in reality, the enzyme would be denatured at a temperature lower than boiling temperature. Usually something like around 65 degrees Celsius, maybe 75 degrees Celsius, depending on the enzyme. The enzyme will be totally denatured. This denaturization can happen with any of these: incorrect temperature, incorrect pH, the incorrect salinity level, the presence of heavy metals, the presence of alcohol. An example of a active protein, not an enzyme, but an active protein would be albumin, the egg white of an egg. When you first crack your egg, it's usually clear and runny and liquid. And then when we cook the eggs, we denature it by heating it. And then the albumin is changed into what was actually a white color, the solid egg whites, a denatured protein. Any question about any of that? If you were to put the albumin in alcohol and then shake it together to mix in the alcohol and the albumin, you would get egg whites. The albumin would turn white as if you had cooked the egg. And it's just from denaturing the protein. The correct temperature influences the amount of enzyme activity. At the optimal temperature, the enzyme has the most activity, and this enzyme has the highest activity at 37 degrees. So this could be an enzyme in a person, at least internal to the body. The pH would be 37. So this enzyme would work best. As you increase the temperature, you start losing the activity of the enzyme as the enzyme becomes more and more denatured. And at this one, at about 50 degrees Celsius, we totally denature the enzyme, and then there'll be no enzymatic activity. Come on. And this enzyme here, when you cool the temperature, you start losing the enzyme activity. And if you cool it down to 10 degrees Celsius, that's refrigeration temperature or below, and there's no enzyme activity at that point. Same with pH. All enzymes have an optimal pH. This enzyme has an optimal pH around pH 5. Where can we see pH 5 in the human body? Anyone? I'm sure some of you have taken anatomy and physiology, so that should help. Your
On the
skin.
stomach.
Say again. No, your stomach would be between pH 1 and 2, at least when the stomach acids are made. So this enzyme has an optimal pH around pH 5. This would be the skin. pH will be around pH 5. And so this enzyme works best in the environment of the skin. Okay. When you move the pH, like start increasing it, like to pH 7, you start denaturing the enzyme. Come on, mouse. And you have less enzyme activity. When you move the pH way high, pH 10, very alkaline, there's no enzymatic activity. And here, when you lower the pH, you also start losing the enzyme activity. And then when you lower it down to below pH 1, down to close to pH 0, you have no enzyme activity. The substrate concentration can also affect the amount of enzyme activity. As I started, stated already, at least when you're low in substrate concentration, when you add a little more substrate, you dramatically increase the amount of the activity to this enzyme. But that only happens up to a point. As you increase the substrate concentration further, this initial increase starts decreasing while the curve starting to decrease. And then once you reach this point, when you increase the substrate further, you get no additional enzyme activity. Why might that be? Why is it that once we reach this point, whenever you increase the substrate concentration, you do not increase the amount of enzyme activity? Anyone? All right, nobody's guessing. It's because
Collision
it's
theory?
well, yeah, it's the
Yeah.
collision theory. But what about the collision theory?
The more substrate there is, the higher the collisions become.
There would be more collisions if you increase the substrate concentration, but then why isn't this continuing to go up? It's staying flat. It's not increasing at all in the enzyme activity. What's happening here is all of the enzyme at this point is already bound to a substrate. So when you increase the substrate concentration, you can't increase the amount of enzyme activity any further because all of the enzyme is already bound. You can't get the enzyme to increase its activity because it's already working as fast as the enzymes will allow it. All of the enzyme, let's say there's 10, all 10 of them are already bound. So if you increase the substrate concentration any further, it does not increase the amount of enzyme activity. Okay. You can also influence the amount of enzyme activity by inhibition. There are two types of inhibition: competitive inhibition and non-competitive or allosteric inhibition. In competitive inhibition, you have a molecule which actually binds to the active site of the enzyme. So normally the substrate binds to the active site of this enzyme, and you add the competitive inhibitor, it can bind to this site, and then the substrate cannot bind to the enzyme. And that's how competitive inhibition works. The two are competing for the active site. Any question about any of that? As I stated, we have some drugs that act as competitive inhibitors, and we're wanting to decrease the activity of this enzyme, if not totally stop it. And you can by adding the competitive inhibitor, which in this case we're saying is a drug, which is only obviously a few drugs. All right, you can also inhibit an enzyme by non-competitive inhibition. A non-competitive inhibition is also called allosteric inhibition. The inhibitor in allosteric inhibition binds to another site other than the active site of the enzyme. So normally there's the substrate binding to the active site. We add the non-competitive inhibitor. It does not bind to the active site. What the non-competitive inhibitor does is bind to the enzyme or protein in another site, and that changes the folding pattern of the protein, changing the shape of the protein, so that now the active site no longer binds to the inhibitor. And that's how a non-competitive inhibitor works. It binds to the enzyme at a site other than the active site. And then by binding, it changes the folding patterns in this protein. So the protein takes on a different shape. The active site is now altered and it cannot bind to the substrate. Any question about any of that? All right, let's talk about feedback inhibition. Feedback inhibition is a type of non-competitive or allosteric inhibition. What we have is a reaction pathway where the substrate binds to enzyme 1, forms the intermediate complex, forming the complex between the enzyme and the substrate, making intermediate A. Intermediate A binds to enzyme 2, making intermediate B. Intermediate B binds to enzyme 3, which makes the end product. Okay, so that's the enzymatic pathway we're talking about. If you have feedback inhibition, then the end product, once it's made, can now bind to the first enzyme, and it binds to the enzyme at an allosteric site that changes the folding pattern of the enzyme 1. So now the active site no longer binds to the substrate. And that's how competitive inhibition turns down the production of the end product. It shuts down enzyme 1. If enzyme 1 shut down, there's no intermediate A, no intermediate A, no intermediate B, no intermediate B. There's no generation of the end product. So what a cell does is when it needs the end product, it proceeds with enzyme 1, enzyme 2, and enzyme 3 to make the end product. And as the enzyme end product concentration increases in the cell, more and more end product will bind to enzyme 1, which will begin to shut down this chemical reaction. When all of the enzyme 1 is bound to the end product, then you'll totally shut down this chemical reaction. And cells use feedback inhibition to regulate how much end product is made. When there's enough end product in the cell, it will then shut down this pathway to stop making end product. Then again, if there isn't enough end product in the cell, then enzyme 1, at least some of them, will not have the end product bind to it. And then enzyme 1 will be active and it'll bind to the substrate, making more of the end product. So what happens to feedback inhibition? Is this end product permanently bound to this enzyme? That's a yes or no. The answer is no, and that is the end product binds to the enzyme 1 as a stoichiomic reaction. So every once in a while, the end product will fall off of enzyme 1. Then enzyme 1 will be active. As long as there isn't enough end product around to bind to enzyme 1, it'll make intermediate A, making intermediate B, making more of the end product. Once we get the end product built up, though, once it gets increasing, it will start binding to the enzyme 1, and eventually you'll shut down all of enzyme 1. When the end product falls off, if there's lots of end product in the cell, then another end product or the same end product can rebind in the allosteric site of the first enzyme to shut it down. If on the other hand, there's not enough end product around and the cell wants more, then this enzyme 1 will be active and will make eventually more end product. That's how feedback inhibition happens. Cells frequently engage in feedback inhibition to regulate how much end product is made and keep it within a level, a range. When there's too little, we'll make more. When there's too much, we'll shut down the production of the end product. Why the end product usually binds to enzyme 1 is you're shutting down the first step of the reaction, and that's the most efficient. If you shut down the second enzyme of the reaction, you do make intermediate A, and that will cost the cell energy. Okay, and so feedback inhibition usually uses the end product, shutting down the first enzyme. Any question about any of that? All right, if not, I think I'm going to stop here and we'll pick up here next time. Any questions? Let me make sure that I've got this at the right time. It should be the right time, but just let me make sure. Come on. Can someone confirm at 6:20?
It is.
All right. All right, I'll see you in the lab at 6:30.